This PowerPoint that you're presenting right now, I don't see it on Blackboard.
I
Am I
sent the
missing?
message out for its location. You need to read your email. Where it is at the bottom left of Blackboard. I'll show you where it is right now.
Okay.
If you go down to the bottom of, if you go to the viewpoint, your viewpoint here, and you look down at the bottom, materials for today's lecture. There it is right there.
Oh, okay.
And
Yeah.
if you go back to the announcements, the announcement was uh, materials right here.
All right.
So
Got it,
it's
sir. Thank you.
incredibly important to read your email.
Yes, sir. Thank
Don't
you.
read you're going to be lost because I make a big effort to do this. So let me ask you to please read your email. But always ask questions. There's no problem. You can always ask questions. So to repeat, facilitated diffusion involves ions and glucose. Facilitated diffusion means that you have a special pipeline or channel, and the flow is from high concentration to low concentration. Osmosis, water moves through the membrane, and water will move from low solute concentration to high solute concentration. So if you look at these dissolved solutes, there's more inside the membrane. So water will move from the low concentration of solute to the high concentration of solute. Okay, let's keep going. So osmotic pressure is a cell can swell or shrink depending upon the movement of water. In some cells, you have special aquaporins, which are special channels that allow water to flow into the cell. A small amount of water is able to pass directly through the phospholipid bilayer. So if you have a high concentration of dissolved substances, the water will move from the low concentration of dissolved substance, glucose, to the side where there's a higher concentration of dissolved substances, glucose. So osmosis can be defined as the movement of water across a selectively permeable membrane down its concentration gradient. So the higher the concentration of water, it's going to move where there's less concentration of water. Volume changes can occur when water moves in and out of cells. That's why it has to be regulated. So, for instance, if you put a red blood cell in isotonic solution, meaning that the concentration is the same inside the cell and outside the cell. So it'll be equal movement into the cell and out of the cell. That's why when you go into the hospital or you need an IV drip, you have to have normal saline. Saline that's the same concentration of compared to the blood, your blood or plasma and so on. If you have a cell placed in a very high concentration of dissolved materials, the water is going to move into a hypertonic. Hypertonic means that the there's a higher concentration of dissolved solute, so the cell will shrink or crenates. If it's hypotonic, there's more water on the outside and more dissolved solute in the cell is going to swell and burst or lyse. So to summarize, a hypertonic solution outside of the cell will cause the cell to shrink. A hypotonic solution outside of the cell will cause the cell to swell. Notice water moves where there's a high concentration of solute. The solute is the blue little spheres that could be glucose and so on. Okay, we don't need to talk about that. Some processes require energy. That's active transport. There are special channels that pump material with energy into the cell from a low concentration to a high concentration. Just know there are 3 different types of pumps: uniport, symport, and antiport. I'm not going to emphasize that much. So one of the most important pumps is the sodium-potassium pump. So what's going to happen is that sodium is going to be pumped into the cell against its concentration gradient, and potassium is going to be pumped, sodium is going to be pumped out, and potassium is going to be pumped in. So it's going to go against its concentration gradient. So this sodium will be pumped out against a large concentration of sodium. This potassium will be pumped in against a high concentration of potassium. So let's do that again. So there's a high concentration of sodium outside the cell. There's a high concentration of potassium in the cell. What the pump does, it pumps sodium out against a high concentration and potassium in against a high concentration. That's why it takes energy, because you have to pump against high concentrations from low to high. So ATP is used to provide energy. So you can see the sodium is pumped out, potassium is pumped in. There's a high concentration of sodium outside the cell. So it takes energy to pump from a low concentration of sodium to a high. It takes energy to pump from a low concentration of potassium to a high. Okay. Sometimes other substances move in with the sodium, like glucose. That would be secondary active transport. Okay, so the outside of the cell has a thin layer of charge. So there's a separation of charge. The inside is slightly negative. The outside is slightly positive because there's an excess of sodium on the outside. Inside the cell, there's going to be a lot of negatively charged proteins and other molecules that can't leave. So this is called the resting membrane potential. The outside of the cell is positive compared to the inside. It creates an electrical gradient. What does that mean? Back to this slide, it means that the inside of the cell is negative compared to the outside of the cell. So resting membrane potential is the charge in voltage for the inside of the cell compared to the outside of the cell. Okay. Vesicles also are able to move substances in and out of cells. And pertinent to today, matter of life and death, the Moderna vaccine and the Pfizer vaccine, the messenger RNA is enclosed in a little vesicle, and that moves it into the cell. So these vesicles are small sacs that can be fused with the cell membrane, and it requires energy. So you can see here, cells can take in a bacteria and eat it. That's called endocytosis. Phagocytosis is cell eating. But when a substance moves from outside the cell to inside, notice a vesicle is forming here. And that's called endocytosis. If the cell eats, it's called phagocytosis. Pinocytosis is cell drinking. So make sure you read through these PowerPoints. I'm emphasizing concepts, but you got to read all of these and study these. Sometimes you have special proteins that help take in substances. It's called receptor-mediated endocytosis. Exocytosis is when a cell in the pancreas is going to release insulin, and that's called exocytosis. I'm not worried about transcytosis. So here's an electron microscope of a substance being released from a cell. Membrane potential. Sodium and potassium have charge. Chloride has a negative charge. Sodium and potassium are primarily responsible for the fact that the inside of the cell is negative with respect to the outside. Current is the movement of ions. The voltage is the difference created by more potassium on the inside of the cell, along with negative proteins and other substances, and sodium on the outside of the cell. So by the cell separates charge, that creates a voltage. So if you look at resting membrane potential as sodium chloride on the outside of the cell is positive compared to the inside. So you can see these negative proteins and phosphate. So the inside of the cell is slightly negative with respect to the outside. Notice there's primarily potassium in the cell, and there's an abundance of sodium outside the cell. So this is what I just said. Sodium, most sodium outside the cell, more potassium inside the cell. This is very important. When channels open, ions can flow into or out of the cell, and that's how nerves and muscle work to move signals. Okay, so here's the concentrations in millimoles. You just got to know that the outside of the cell is positive with respect to the inside. Okay, let's go stop the recording for a moment because we're at 15 minutes. Stop recording.